All right. Give me 1 second here. Holy, good evening. What's up, Grouchy Beer? What's up, JP? Captain, SVC, how's it going? JP, the Koi Pit, how's it going? I don't know what rotifers are, so I'm going to learn a lot. That's the goal. Hopefully, we're all going to learn something. Hold up. Give me one moment. One moment. What's up, Mick? How's it going? Beto, I saw Beto up in here. Diana, how's it going? Let me turn this water on real quick. I gotta finish one last water change. My days, my workflow has been devious recently. This is going crazy. I'm not gonna lie, stream was a little late. I had to take a nap. Bro. I was exhausted. That's why I wasn't ready for stream in time, because I fell asleep. I ain't gonna cap with you. I fell asleep. The thing is, this is kind of how my schedule's been going. So last evening, I do all my water changes and stuff. I catch everything. I gotta ship some stuff. I catch everything the night before, especially when it's fish. I try to catch it the night before so they void everything. Then I wake up this morning at 5. Then all the things I caught, put it in fresh water. I pack up all the inverts, but I saved some fish. So I wake up at 5, I pack all the stuff, I go to work by 8, then I come back, rush back, so I can pack the fish in fresh water again, the fish I left, and then make it to the post office by 5. Oh my gosh, bro. It was such a ridiculous time to fit in there. But it's definitely been challenging since my work schedule has changed to basically be the most efficient schedule for me to have a hard time making it to the post office. So basically, I'm waking up 4 or 5 a.m. to pack stuff, and then either I have to try and get home quick enough to take it to the post office myself. I get off at like 4:30, or I gotta find someone to drop it off for me. What's up in the flesh? How's it going? Big Steve said, did the hardest working say he was sleeping on the job? Well, no, not on the job. Come on, don't be ridiculous. Not on the job. There's just always stuff to do in the fish room, man. After the stream ends, I'll be in here for another 2 hours. But been getting a lot done. I got one lonely Glow Daniel fry randomly. But I found my first eggs. They finally started laying eggs again. So I got finally gained some eggs from those. But then there's the fry just in the tank with the adults, man. Just cruising at the top. So I want to talk about rotifers today because I kind of roughly know what they are. I was reading some stuff about them recently with some usages and culturing different fish fry and stuff. And I'm trying to get back on my live food game. I've actually been all right on the worms, the micro worms they're going well. I need to make some fresh cultures actually. The grindle worms are doing all right. The white worms, we don't need to talk about it. But I want to get back on my hatching brine shrimp and stuff like that. So I'm kind of back in the reading about different live food cultures. I've been doing the peanut beetle larva. I've been mostly, since it's such a pain comparably to harvest the peanut beetle worms, larva, whatever, I'm mostly just target feeding them the specific things. So like pea puffers and the Congo puffers, I'm giving them those because, it's a nice nutritious worm. I only need to get a certain amount for those. So also, they're not all the fish respond to them that quickly. They don't have the best movement in the water. So that's another thing with the peanut beetle larvae I don't like, because they don't move around that much for long enough. But yeah, so I'm kind of on that frame of mind, and something came across my face, my eyes. Yeah, the two eyes that are in my face. I guess my face is accurate. Yeah. Something came across my face about the mass culture of rotifers, and I was like, well, maybe now's the time that I actually try and remember what the hell these things are. And as per usual, that means I'm gonna make it all your problem. We're all gonna have to try and figure out, should we try and culture rotifers? Is there any reason at all to culture them? Catton says, my opinion, rotifers are difficult to culture. Paramecium is easier for me to keep. I think that could be a consideration. Although I haven't really. I think my problem with paramecium is I haven't really had to use it, so it's kind of hard for me to keep up with it because I don't really feel like I rely on it that much. Cat says copepods and hydra predate on them, so you have to be careful about contamination. All right. JP said almost got it with the black T-shirt. Oh my, JP, you guessed my outfit again. It's getting scary, man. you in my freaking in my freaking closet. What's up, She Savage Cichlids? Steve said, what kind of glow Daniel? Sorry, not glowfish. The glow light Daniel. The Chopra. What is it? What's the Latin name? Daniel Chopra. Glow light Daniel. I'll put a photo on the screen. By the way, great fish. Not very expensive, but it looks fantastic. It really does. So I recommend, if you're looking for a fish that is very active, Glow Light Daniel is kind of a nice pick. One second, I got a picture of it on the screen. But I'm trying to get them breeding. I think Lowell has a video about breeding them. But good looking fish. Not gonna lie. But that's what a Glow Light Daniel looks like. Let's start getting into this rotifer thing. Smart Person says rotifers are fine, but intentional culturing is tricky. Straining in a 53 micrometer mesh seems to work with isolation, but look at it funny, it could become contaminated. That's one thing I'm hoping we get through today. Possibly there's some good methods we can take a look at on how to better culture these things. Perhaps keep the contamination to a minimum. But we got to start off with the basics. I think we've done a good job the last few times we've read something of building up to where the hell we're going. And what better place to start than just simply what are rotifers? So with that being said, let's get into it. What's up, Jonah? Jonah GLA. Jonathan said, I think Chopre is in Daniel again. I think it is. It's select. Oh my gosh, we're gonna have to look this up. Let's see. Glow Light Daniel. What's the best? Let's see. Wikipedia. Daniel Chopre. All right, that's one. I see Select These Chopre and Rachel O'Leary from 2016. Then the Aquafish has Daniel Choprai instead of A-R-A-E as a R-A-I. Man, what's going on with the Daniels these days, man? Aqua Import still has them as Silistixes Chopre. Alright. Redescribed. When did this happen? What happened here? Well, it's like you start off with a freaking sidestep already. I like how the Wikipedia page for this starts with, this should not be confused with glowfish. People really have a problem, like, isn't that funny? People have a problem identifying the difference between a fish just called Glow Light Daniel and glowfish. Like, one is manufactured, one they just call that. Pedro says, a professor's associate who I emailed said they use cultured rotifers to feed green spotted puffer fry. Yeah, I got a few things pulled up, some different case uses for them. So I think I'm glad to see that people are as intrigued by the rotifers as I was. Now, let's start with what a rotifer is, because this is what I'm gonna need. I'm gonna need to really just go step by step here for my own benefit, really, before we even get to the mass culturing of them and all that kind of stuff. I need to actually think about what the hell these things actually are. So here's a picture, and we're back to the whiteboard. Thank God. I got the whiteboard back up. Here's a picture of a rotifer. And we have a nice little measurement here. 10 micrometers. Now, I'm going to do 10 micrometers. That's 0.000393701 inches. All right, just for, comparison. Let's see. Do I have 10 micrometers banana for scale? Like, is that a thing? Wow, they haven't done that conversion yet? I'm just ahead of the game. Oh, wait, we got banana for scale.info. All right. Let's see. Do we have micrometers? Yeah, they don't really seem to be up on this for the banana scale. I'm going to have to submit a complaint. My apologies. Parna said, aren't rotifers used in saltwater? I'm confusing stuff. There are some saltwater. They are used in saltwater a lot. Savage Cyclist says it looks like a gummy bear to you. I could see the gummy bear comparisons a little bit. I could definitely understand that. Give me 1 second. I'm finding my page real quick that I got this photo from, so we can get into it. Now, another thing I want to do, as we're talking about the rotifers and what they are, the uses of them, and all that kind of stuff that we can glean, I also want to talk about them in comparison to things like brine shrimp, because that is definitely why do this if we can just hatch some baby brine shrimp? It's got to have a unique place. Now, this first paper that I'm taking the definition of what the micrometers are. And Captain's Critter says, oh, I'm overflowing my tank. Oh my God, I got distracted. 1 second. Oh my God. I literally just flooded the tank right now, live on stream. Give me 1 second. It's all good. I caught it right before it really became a big issue, so I think it's all good. I say that, and there's water all over the floor. All right. Well, that should be good. Now that is mitigated, it was the nap that did it. I'm still waking up. All right. My gosh, I think that should be good. Let me point the fan over there. Oh, it is. Okay. All right. So the paper I'm gonna pull this first definition from is actually, someone mentioned the saltwater stuff. Actually kind of appropriate, because this first one I'm going to read from is The Importance of Rotifer as a Live Feed in Mariculture. Now, of course, as any good paper does, we got all sorts of photos in here. So let me read a few sections to get introduced to the rotifer. So in the introduction here, it says, fisheries and aquaculture remain an important source of nutritious food for millions of people around the globe. Yep, setting the stage for why it's important to raise some fish. All right. However, larviculture of most species remains the bottleneck in establishing full cycle culture. Here we have a figure of a hybrid groper cross. Kind of ugly, not gonna lie. Success in aquaculture is always linked to success in seed production, and it hinges on a deep understanding of the early larval stage of the cultured species. Larviculture, or the first feeding of early larval stages, is apparently the major bottleneck for the professional upscaling of the culture of marine fishes. So we got some freshwater focused stuff later, but this is just kind of, let's get introduced to these things. First feeding is a critical stage in larval development of many fish and aquatic organisms. Yes, of course. During this stage, fish larvae require a live feed with suitable nutritional value and a suitable size range for the success of larviculture of many fish species. The size of the initial feed should match the size of the larva's mouth. It should be small enough to be ingested by the larva. So, here we come in with the rotifer. Now, this rotifer they're showing us is Brachionis picotilis. Yeah, picotilis. And this is observed under a light microscope. They describe the rotifer as a minute, unsegmented, pseudocolumate aquatic invertebrate that has a round and flat spineless body. Rotifers are widely distributed in freshwater lakes and ponds, as well as in marine water. Approximately 2,000 species have been described. The phylum of the rotifer is known as Nemathelminthes or Aschelminthes, and it belongs to the class of Rotiferia, order Monogonata, and the family Brachionidae. Initially, rotifer was all classified as one species, etc., etc. We're not going to get into the weeds of all that. The size of the male rotifer is much reduced and less developed than the female rotifers. Interesting to know. The body of the rotifer is generally covered by a distinct cuticle and has a bilateral symmetry and sexual dimorphism. The body is comprised of a head with a corona, and trunk and foot. The head carries a rotary organ named the corona, which is easily recognized by its annular ciliation. The corona assures locomotion and whirling water movement, which facilitates the uptake of small food particles. The trunk contains the digestive tract, a mastax that grinds the ingested particles, the oesophagus, the stomach with gastric glands, and the intestine. The foot is an appendage that extends from the body ventrally with 1 or 4 toes. Damn, the rotifer's got toes? This is opening up all sorts of doors. Here is a figure showing the diagram of the life cycle and reproductive pattern of the rotifer B. plicatilis, showing the asexual and sexual reproduction, so they can do both. DP Exotic says they remind me of water bears, the tardigrades. I could see that. Smart person said rotifer feet pics win. I mean, I guess the foot has to be, what, this thing here, perhaps? It says it extends out. 1 second. The foot is an appendage that extends from the body ventrally with 1 or 4 toes. So it probably is this, right? So here's how they reproduce. It's known that the rotifer can use 2 modes of reproduction, asexual and sexual reproduction. The reproduction involves a mycetic diploid 2 N chromosomes and mictic haploid N chromosomes females. All right, parthenogenesis happens in the amictic females, which produce amictic eggs. These eggs are later developed and hatched into females. So females hatching more females. Generally, rotifer undergoes asexual reproduction when there are no environmental threats, such as changes in temperature, salinity, and feed density. It's kind of like, reminds me of the Daphne and stuff. The mictic females will switch to mictic females during sexual reproduction under specific environmental conditions, where the mictic females produce mictic eggs and develop into haploid males with a size about 1/4 of the female. The mictic males and females will undergo sexual reproduction and produce resting eggs. So this is kind of a familiar strategy that we've encountered a couple times at this point. Let's get to the nutritional value. In marine hatcheries, rotifers are the most suitable live food for rearing marine fish larvae. They're commercially available, and the production as feed for marine fish is not complicated. However, there are disadvantages to using rotifer as live feed. One of the advantages that make them unsuitable as prey is that they are not easily digested. Besides that, their swimming movement does not encourage feeding response in all marine fish. That's a marine fish. The nutritional value of a rotifer depends on the feed that's given to them. The protein content of a rotifer ranges from 28% to 63%, and lipid content from 9% to 28% of dry weight. The carbohydrate content ranges from 10.5% to 27% of dry weight. And the lipid content of rotifer unequivocally has the greatest influence on the growth and survival of marine fish. Ecosapentaenoic I tried to just fake this one to get it off. Ecosapentaenoic acid and DHA of the fatty acid components and lipids are known to be essential for the survival of marine fish larvae. So usually the rotifers, they do enrich them for EPA and DHA content. I thought this was, I got some other culture systems to get to for their freshwater uses, but I thought this was interesting. They said the rotifer was first considered as a pest in eel culture ponds in the 1600s, which caused damage or rapid change of water color. When the bloom of rotifers occurred, the supply of oxygen became insufficient for the eel that suffered motility due to suffocation. However, they were soon discovered as a potential first feed for freshwater and marine fishes. 1979, and mass production of the rotifer was first developed in Japan in the 1960s, and 25 years after the first production of rotifer, several culture systems began using them. In rotifer mass productions, a few problems must be considered, such as the rotifer strain selected must be appropriate for the larvae to consume. Now I'm already starting to get scared. I gotta pick the species of rotifer. Mick said Quentin Tarantino will love this. Oh, the feet. Probably. The water quality, feed, and quantity must be maintained to an adequate level to ensure the survival of the rotifer. In addition, suitable cultural conditions that are convenient for the hatcheries must be considered as well. There are 3 types of rotifer culture systems. Write this down. Batch, semi-continuous, and continuous cultures, which are commonly used in most hatcheries. What's up, Beto? How's it going? Beto said, what's up, ISO peeps? ISO peeps. That's a new one. I like it, though. I like it, ISO peeps. So there's 3 types of rotifer culture systems: batch, semi-continuous, and continuous cultures, which are commonly used in most hatcheries. Each of the culturing systems has advantages and disadvantages, and a selection is according to the suitability for hatchery management. So let's get to the first type, the batch culture. Most of the efforts made for the culture of rotifer were in the batch culture system, where microalgae raised in a large tank were used as a primary diet for rotifer. Baker's yeast has also been used as a food for rotifer, but yeast is lacking in a few nutrients essential for rotifer and fish larvae. Due to this, different direct and indirect methods of rotifer enrichment become necessary before feeding the fish larvae. Now, it is the main challenge for scientists to develop a stable and sustainable high-density culture technique of rotifer, which will reduce the use of hatchery space for live food culture, increase larval production, and reduce the cost of production. The batch culture system that harvests totally all the rotifer, with part of the harvest used as feed and the other part as inoculum for the next batch cultures. This culture has some disadvantages, as it can cause highly variable conditions and growth performance in terms of the biochemical composition of the rotifer composition due to the unstable physiochemical water parameters. Next, we have the continuous culture. Continuous culture controls the feeding, water quality, and density of culture organisms. So you kind of see, I don't know if you got it before, but it's a batch culture. You know, you get you put them in a huge batch, a big tank with microalgae. You know, they kind of have the big boom. You harvest a bunch. You harvest all of them, and some of them you feed, and some you keep for the next. So you're doing different batches. This one is continuous culture. Continuous culture controls the feeding, water quality, and density of cultured organisms, and this is also known as the feedback culture, where a certain volume of rotifer is harvested daily from the culture tank. So not just like they're harvesting them all in one go. Continuous culture offers easy manipulation of rotifer physiological and nutritional quality. The system produces a log phase rotifer that can be harvested continuously. Nutritional quality is maintained by providing adequate feed. However, the initial cost of the culture is high due to the use of an automated control system. That's kind of cool. You can automate it. Aquatic says, why go through the trouble and not make it continuous? Well, I think what they just said there has a high initial cost. Koipit says, I have a question. Does the research study disclosure where the study's funding comes from? Usually they do have at the end somewhere there's some kind of conflict of interest thing. This one is actually from a book. This is actually an excerpt from a book. And the book is called Essentials of Aquaculture Practice. And let me see. It's a textbook. It's a Springer textbook. So this one is a little bit different than, it's not like a typical paper. It doesn't have like a funding thing. This is just for learning, I guess. Damn, how much does this book cost? Let me see. $154 for Kindle? Oh, hell no. I'm reading this for free online. What's going on? I'm reading this for free. Okay. I got the exclusive. No, I'm joking. Okay, let me go back to where I was. That's so funny. Okay. As I was saying, good question, though. Good question. All right. Continuous culture. So, yes, it has a higher upfront cost. Got it. The continuous culture, because it has an automated control system. The continuous culture method can be seen to have high daily production with stabilized water parameters that can be achieved by chemostat or turbidostat. All right. Let's look up chemostat. Chemostat. Okay. A chemostat. A bioreactor to which fresh medium is continuously added while culture or liquid containing leftover nutrients, metabolic end products, and microorganisms is continuously removed at the same rate to keep the culture volume consistent. Wow, we got a cool graph here. So inflow of fresh sterile medium comes in, so water. We got sterile air, everything's sterile here. We got flow rate control for the water. Air opening, gaseous headspace, the culture, so I guess the rotifers would be here, stirring apparatus, and then the outflow of media and microbial cells to collection vessel. All right, I'm following that. When the culture, let's see. Yeah, chemostat and the turbidostat. How's the turbidostat different? It's also a continuous microbiological culture device similar to the chemostat or an oxostat. Oh my gosh, this keep coming on new ones, which has feedback between the turbidity of the culture vessel and the dilution rate. The theoretical relationship between the growth in a chemostat and growth in a turbidostat is somewhat complex. Well, that's where I stop. Okay, that's another stream. The chemostat system allows constant flow rate of culture water with a single component controlling the growth rate of culture. This causes the water parameters to be more stable and produce a favorable environment to produce a higher density of rotifers. However, for the turbidostat system, there is no constant flow. However, water is changed in a period, and the specific growth rate is controlled internally by the culture reactions. Rotifer culture is successful when microalgae diets provided are sufficient and can sustain the rotifer to avoid a culture crash. So we've covered batch. They set up the culture. Population booms, they scoop them all up, feed some of them, keep some to start the next culture. Ooh, I'm in live chat. And then continuous cultures. So they got the water changes flowing. They're just harvesting them every day, keeping it going. Aquatic says, so how often is the water changed at that point? I think with the turbidostat, it might be more variable. The chemostat, it's constantly flowing sterile media in. The semi-continuous culture method is a combination of the batch culture and the continuous culture method. A certain volume of rotifers harvested daily from the culture tank. This is like the continuous culture, but the rotifer is harvested according to the batch culture during tank changing, which is called the thinning system. The thinning system, okay. This culture is usually conducted in larger tanks, such as 50 to 100 tons, and it's always maintained at low densities. The disadvantage of this culture system are the requirement for a larger area in the hatcheries for culturing and the excretory products, solid waste, and nitrogenous products that accumulate in the tanks that lead to their collapse. This sounds terrible. The selection of a culture method is critical to rotifer mass production. In comparison with batch culture techniques, semi-continuous cultures are considered more promising and reliable for stable and cost-effective rotifer conduction. Chemostats are designed for constant input of limiting nutrients, by which the growth rate of cultural organisms is maintained constant. In contrast, turbidostats are continuous culture devices where the population density of an organism is held constant by continually adjusting the washout rate or harvest rate to offset the variations. Both chemostat and turbidostat cultures offer easier manipulation of rotifer physiology and nutritional quality. All right, so here we have a schematic. Wow, they didn't give us the schematic of the other systems. We had the schematic of the semi-continuous culture system of the rotifer. So this one is for a salty rotifer. Deep said, I've been thinking of trying to do a super culture for feeders that like the aquarium co-op video with black worms, red neon shrimp, daphnia ammonium, and parameciums. I think these guys might work in that. Man, you're throwing in everything. I wonder if the daphnia eat rotifers. So here we have the water coming in with the filter tank. Water coming in, filter tank. It looks like a 0.1 micrometer filter, perhaps, the water goes through. And my tank said, Captain said, can I share the party mix link? Yes, you can. Shamelessly plug yourself. Captain's Critters has some great live food cultures, including if you're looking for that photosynthetic red bacteria. That's a good one. Captain said, yes, Daphnia will eat rotifers. Oh, damn. But share your link away. All right, so water comes in, goes through the 0.1 micrometer filter situation. And by the way, Captain's Party Mix, I think it's 30 ounces with Daphnia, Magna, Moina, Copepods, Paramecium, and Infusoria. It sounds like a party as long as you don't drink it. Put it in a tank. Don't drink the party mix. All right, so after it goes through the filter, and the diamonds, these are pumps. So pumps it through this filter, goes into another tank that pumps it up through here. This rotifer culture tank has a heater in it and a rotifer filter, and then another pump. So rotifers come in. Sorry, water comes in. We got the rotifer culture, the filter. This pumps it over to the rotifer harvest, which just has a heater in it. And there's air stones in the rotifer harvest and the rotifer culture. And then their food supply is put into the rotifer harvest. So they're coming in here, then they're coming into the harvest. It's pumped. The food supply is pumped into the rotifer culture and the harvest container. TP said, I've been looking at your site, Captain. I also want your green water culture. Oh man, let's go. Diet and enrichment types. Okay, here we go. The nutritional quality of rotifers is crucial for the health and growth of the larvae of marine organisms. So we got yeast. They use yeast as a substitute diet. However, culture fed with yeast is sometimes subject to sudden, unexpected decrease or suppressed growth of population. Vitamin B12 is an essential vitamin for rotifer growth. And previous studies show that rotifer grows better at low oxygen levels when fed with yeast because the bacteria in the culture water produces vitamin B12. Well, that's interesting. They're facultative anaerobic bacteria. The bacteria in the culture water, due to the suspension feeds and animal excretion products, also becomes feed in a rotifer culture. However, excess food and excretion products tend to pollute culture water by increasing ammonia levels, which limits the population growth of the rotifer. Excuse me. Steve says, my green water culture has been pumping. Is the red stuff fresh water? I think the red stuff is Yes. I think also there she's got the marine and freshwater green water. Microalgae. Microalgae known as phytoplankton occur in a vast range of sizes and forms. I think some people feed a mono or try to feed a mono shrimp babies rotifers too. I thought I should mention that. I think that's another use that I've heard people try and use them for. If Daphne eat them, then probably the baby amanos can at some point. So we're feeding them microalgae. All right, let's get to the next thing. Captain said, I'll keep you updated when back in stocks. An acquaintance purchased 600 ounces of the stuff, and I'm still recovering. Oh my, 600 ounces of the red bacteria? My goodness. Someone should do a wellness check. Where are you going with all that? Where are you going with all that photosynthetic bacteria? All right, let's look at a research article on using rotifers and their impact on the culture of the Florida bass. All right, not quite an aquarium fish yet, but let's look at one case. This is from the Aquaculture Research Volume 2025, Issue 1. I'll put the link to this in the chat. I like to, for the live food stuff, I love getting just the aquaculture articles. They got the most money on the line with this stuff. Captain says, red PSB is freshwater but requires some sodium to thrive. All right. Thriving bacteria. Here we are. So this article is called Rotifers Improve First Feeding for Indoor Culture of Florida Bass, Microterus salmoides. If you're not someone that fishes a bunch, which I do not, you just don't know every single fish out there, I'm going to pull up a picture of the Florida bass. All right. Well, let me make sure I get an actual picture. Microterus, let's go to the freshwater. FWS.gov. So they're all over the damn place, huh? Largemouth bass. Big old ugly fish. Holy. I need to see it. Let me see a video of these guys. Let's see. Yo, I don't need an ad. I'm getting an ad for Depop. Come on. I'm not that tired of GP guessing my wardrobe. Alright, we have a guy holding the fish. Yep, this is educational for me. So this is a fish that exists. Perfect. I'm glad I've made my point. In the intro, it says largemouth bass is arguably the most sought-after recreational black bass species in the United States. Production of this species in North America dates back to the 1930s with the goal of supplementing natural populations. Okay. High demand largemouth bass. Damn, that thunder was loud as hell. However, largemouth bass and Florida bass has recently become the focus species for aquaculture as food fish. These fish possess high tolerance to stress, fast growth rates, and excellent meat quality. Pause. God damn. In 2022, global largemouth bass and Florida bass production was dominated by China, which raised 800,000 tons of largemouth bass and Florida bass, equating to over 99% of production. In comparison, the U.S. produced 1,930 tons of largemouth bass. That's pretty pathetic. The top 5 cultured species in the United States behind catfish, channelfish, and the channel X blue catfish hybrid, trout, tilapia, and hybrid striped bass. Among these species, largemouth bass and Florida bass rank highest in market price for food size at $7.53 per pound, with catfish, trout, tilapia, and hybrid striped bass averaging $1.2 to $4 a pound. Okay. Arguably one of the most critical... Sorry, 1 second. Let me read this. I skipped over a part I think is good. Broodstock are first brought from outdoor earthen ponds to an indoor facility where they spawn on spawning mats. Following fertilization, spawning mats are brought to the nursery ponds where fry will utilize pond productivity until they reach the size of 38 to 51 millimeters. Fingerlings are transferred to an indoor facility where they undergo feed training on commercial diets. Finally, feed-trained fish are stocked in grow-up ponds until they attain market size. The entire process can take up to 2 years, which is one of the reasons the species commands such a high market price. The success of this procedure is highly variable due to the fluctuating environmental factors, exposure to predation, and unpredictability of nursery pond productivity. Koi said, "What do the crab shells do?" Wait, when did I what crab shells you speak of? All right. 2 years to get this fish to market size. Damn. The success of this procedure is highly variable due to fluctuating environmental factors, as I read before. Therefore, transitioning production to an intensive indoor recirculating aquaculture system, or RAS, could eliminate the uncertainty of pond productivity and allow more efficient feeding techniques and better control of water quality parameters. Arguably one of the most critical periods during early life history is the transition from endogenous to exogenous feeding. Yolk provides nutrients necessary for organ and tissue development, formation of the digestive system, and anatomical features aiding in prey capture. Once endogenous resources have been depleting, timing of exogenous feeding is crucial, where poor transition to exogenous resources have been linked to slower growth rates, nutritional deficiencies, deformities, and lower survival. While some species, rainbow trout and channel fish, can be successfully raised on artificial diets from first feeding, largemouth bass require initial live prey before accepting artificial diets, including the intermediate code feeding period. Lost to Fin America said, "Hello everyone." What's up, Lost? How's it going? So here comes in the rotifers. Rotifers and Artemia are the two most common live feeds utilized in larva culture. Unfortunately, culturing live feed is labor intensive and has high production costs, is vulnerable to culture collapse, and can be a vector for disease. Additionally, their application remains challenging due to the variable quality in size, hatching rate assists, and fluctuations in price. Many studies have focused on the early diet regimes of other warm water species, but there's limited information on the capabilities of largemouth bass. Alright, so let's see how they did with the rotifers and the Artemia. So materials and methods. Now we're reading a real paper. We got materials and methods. Let's see what we think of this one. And I'm sure there's got to be a conflict of interest in here somewhere. Unless is this a university paper? National Institute of Aquatic Resources, Auburn University, alright, Technical University of Denmark, Department of Biology of Mount Island. Okay, this is like a university thing. Alright. Animal care protocols for fish experimentation were reviewed and approved by Auburn University Animal Care and Use Program. The Florida bass were raised in concrete raceway, 27 by 3 by 1 meter at Red Hills Fishery in Boston, Georgia. They even put the coordinates in here, where they were fed live goldfish to satiation. Spawning was induced by following a 10-hour photo period for 4 weeks, followed by an 8-hour photo period for 3 to 4 weeks, a 10-hour photo period for 2 weeks, and finally a 14-hour photo period for 2 weeks. Spawning mats, Spontex Pentair Aquatic Ecosystems, the brand they used, were utilized and evenly distributed along the raceway during the spawning event. This is a spawning event. Florida bass broodstock were represented by 42 males and 51 females. Length and weight range from 324 to 470 millimeters and 0.49 to 1.87 kilograms respectively. The spawning season took place from 15th of October to the 31st of November 2021, where water temperature and dissolved oxygen ranged from 19.4 to 23.1 degrees Celsius and 8.15 to 10.23 milligrams a liter respectively. Mean nitrite was 0.019 milligrams a liter, and mean nitrate was 0.8 milligrams a liter, and mean total ammonia nitrogen was 0.07 milligrams a liter at embryo collection. So embryo collection you're learning, we're just getting through the methods of how they measure this, and we'll get to what the results were. Embryo collection and rearing. Embryos were transported from Red Hills Fishery to Auburn University on the 15th of November in 2021 in 114-liter coolers that contained 40 liters of raceway water. In total, Florida bass embryos were represented by 11 spawning mats. At arrival, spawning mats were suspended 15 centimeters below the water surface in a 25-liter aquaria. The water temperature was set to 21 degrees Celsius upon embryo arrival, which reflected the water temperature of the coolers. At peak hatch, the water flow was adjusted to slowly raise the water temperature to the desired steady temperature of 27 degrees Celsius. 2 recirculating aquatic systems were utilized, with each system containing 8 aquariums. Each RAS contained a UV filter, bead filtration system, bag filter, 0.5 HP pump, 3 795-liter circular blue tanks, 2 190-liter sump tanks, a heat pump or inline heater. In addition, systems were equipped with diffused air and a flow rate of approximately 7 liters a minute. Temperature and dissolved oxygen were checked twice daily. In addition, other water quality parameters were tested twice weekly using a spectrophotometer. Nitrate and nitrate levels were kept between 0 and 0.02 milligrams a liter for ammonia, 0 and 0.05 milligrams a liter. That's for ammonia. Nitrite, the first one was for nitrate and nitrate. That's kept to 0 to 0.02 to 0.02. My goodness. pH is 7.5, 7.7. Alkalinity 95 and 125 milligrams a liter. And hardness 80 and 90 milligrams a liter. Okay. So here's the diet regimes for the different batches of the Florida bass larva. Ben said, "Can I have a free fish take?" What? They silenced Ben asking for a free fish take? How dare they? How dare they? Before the stream, I thought they were just plants. Oh my gosh. All right. Here we go. So treatment 1, they fed rotifers plus enriched artemia. Treatment 2, we got rotifers plus non-enriched artemia. In treatment 3, enriched artemia. And treatment 4, non-enriched artemia. So we got the rotifers in addition. There doesn't look like there's anywhere rotifers on their own, because that seems like they're always going to use brine shrimp regardless. But they're seeing what adding the rotifers in does. Okay. Let's see. The Florida bass were fed 6 times per day with rotifers and premium grade A Artemia napoli from Brine Shrimp Direct. They were fed Autoheim Micro Diet. That's from Japan. And Shredding Starter Feed, or combinations of these diets. In total, there are 4 dietary treatments with 4 replicate aquarium per treatment. Okay, 4 replicates per treatment. I'm looking up this diet. Okay, it's just a small little larval fish food. Alright. In total, there are 4 dietary treatments. Treatment 1 received rotifers with enriched artemia. Treatment 2 received rotifers with non-enriched artemia. Treatment 3 only enriched artemia, with treatment 4 receiving only non-enriched artemia. Artemia were enriched with RRI N3 at 0.35 grams a liter, 50% protein, 17% lipids, and 68% moisture. They put HUFA and DHA from shredding in there. And at peak hatch, and every 12 hours, starting at 0 days post-hatch, larvae were fed 2 Artemia milliliter and 5 rotifers a milliliter. At 5 days at peak hatch, all treatments received the micro diet that I just saw at 0.5 and 1 gram a tank per feeding. The rotifer feedings were reduced by 1 milliliter daily to slowly wean the Florida bass off of rotifers, starting at 8 days peak hatch. By 12 days, the fish were weaned off rotifers. Artemia were increased to 5 milliliters at 8 days to supplement to rotifers reduction. From 9 to 12 days, Florida bass were transitioned from the smaller fry food to a larger version of it. Rotifers were cultured in a 190-liter conical tank at 23 degrees Celsius and a salinity of 7 ppt. All right. They're fed 6 times per day at a rate of 0.5 grams for 1 million rotifers. Photo period was set for 24 hours. Artemia was cultured in 12-liter hatching cones at 26 degrees Celsius and a salinity of 35 ppt. To ensure proper prey densities, 1 milliliter samples were taken daily from the rotifer and Artemia cultures and counted in duplicate. Rotifer culture followed the batch method. There we go. See, this is why we set I know what the batch method is. We did this in order. Rotifer culture followed the batch method, where approximately 170 liters of culture were harvested every 5 days. Artemia were cultured for 24 hours, collected using a mesh screen, and rinsed with distilled water before feeding. Then this is how they collected the data. This is going to get, you know what's about to happen to the data. Fish that are data, it doesn't end well for them. Fish were sampled at 2, 4, 8, 12, and 20 days, and 24 days. For each sampling, 20 random individuals were selected from each replicate. For this study, 0 DPH was defined when more than 50% of eggs have hatched, and you already know what happened next. The fish were euthanized with 150 to 200 parts per million MS-222, tricane methane sulfonate. It's branded from Argent Laboratories. Wow, my favorite company for euthanasia of fish. So they sampled 20 fish at those day intervals, slaughtered them, and then observed them using a Zeiss stereo microscope equipped with 0.5 to 1x objectives and Zen 2.5 imaging software. What's up, Johnny Best? How's it going? Don't tell Dan. That's hilarious. Measurements were conducted. Okay, they looked at it. So let's look at what went down here. We have all the graphs and all the numbers. Give me a second to take this in. So here's the different treatments. TL equals total length, how long the total length of the fish. ED equals eye diameter. That's an interesting thing they're measuring. DFN is a numerator, degrees of freedom. DFD, denominator, degrees of freedom. MH is mytome height. JL is jaw length. BA is body area. YA is yolk area. And ODA is oil droplet area on this graph. Yes, Steve, they got to wean them off the rotifers onto the manufactured food. Now, I got the graphs here. We'll come back. We see here no rotifers and with rotifers on total length. So this graph shows the total length and the no rotifers versus rotifers. It's pretty close, honestly. At the end, the ones with rotifers are a little longer. And okay. Over here, I think these are all the, yeah, there's the mytome height. Oh, this one, the rotifers are a bit significantly larger there. And the jaw length by 24 days, the rotifer is significantly larger there. So there's a noticeable difference. Hey, thank you, Johnny Best. W Johnny in the chat. Johnny gifted 1 membership. DP Exotics got it. Nice. Tomorrow I gotta do a members only stream. It's the only way. That's what I'll do. All right. Let's look at the results here. Whew. Okay, this is what they use to calculate the results. There's a significant impact of rotifers for most morphological traits. I just read all those ones above. At each age, where fish increased in size when offered the rotifer diet. However, as no higher order interactions were significant, the dietary regime and main effect were interpreted. Here, Florida bass were all in better condition when fed the rotifer diet, and overall, Artemia enrichment did not significantly affect any of the morphometric traits. The age x rotifer interaction was significant. Therefore, these saturated models were decomposed into separate t-tests at 2 and 4 dpH, where both yolk traits decreased when larvae were offered rotifers. Here, larva-fed rotifers utilized their yolk at the fastest rate and were most efficient at converting their yolk reserves to body size compared to those not fed rotifers. On the other hand, Artemia enrichment did not significantly impact any of the yolk-related traits. While neither rotifer nor Artemia enrichment significantly impacted the Florida bass weight, final biomass, or survival up to 25 dpH, 25 days peak hatch. All right. Here I'll pull up the graphs here. Let's see. Yolk area. So this is the one for the yolk area. So they're saying the ones that they fed the rotifers were able to more efficiently convert their yolk, use it up. That's kind of interesting. So there's one little case thing. I put the link in the chat one more time. I put the thing in the chat one more time. Let's look at another culturing of rotifers. We'll do this bounce back and forth thing where we look at a culture system and then we look at a result someone got eating the rotifers. JP said, "Where's Mick?" Mick has been watching the World Cup. Let's be real. Okay, let's be real. That's where Mick's at. There's probably a World Cup game going on right now. Let's take a look at this automated system of tubular filtration for rotifer production. Come on. Come on, you know that's what you want to hear about. Okay. I'll breeze over the introduction. Featherfin says, yeah, the U.S. is playing also high neck. What's up, Featherfin? Yo, Featherfin tried to hack me, bro. He tried to hack me. He sent me a Discord DM. It said something like, what did it say? Oh, he deleted it from the... It was something like, buy this crypto. Man, he tried to take me out. What's going on? All right, let me breeze over this introduction. This is called... I read the title and I put the link in the chat. We're good to go. So rotifers, essential organisms for promoting fry growth, primarily from days 3 to 20. For instance, in grouper fry breeding. This typically constitutes the first exogenous feed offered to marine fry when their yolk sacs are exhausted. Featherfin says, I downloaded a sketchy ISO mounting program or something. Gotcha. Mick said, hey JP, I'm here, just a bit tired, back after 4 days caring for old mom. Rest up, JP. Rest up, Mick. It said, rotifers are rich in essential fatty acids, including highly unsaturated fats, etc. Just kind of going over why they use them. In commercial aquaculture, the water used for culturing rotifers often contains relatively large copepods that originate from unfiltered natural seawater. Oversized copepods generally cannot be swallowed by fry. Feeding rotifers with algae, fungi, and organic matter during the culture process often results in the production of large quantities of nitrogenous waste, which increase the ammonia level and toxins and substantially reduces the oxidation potential of the culture medium. When a rotifer culture solution is directly added to a fry-containing pond, the water quality of the pond deteriorates, which negatively affects fry growth. Moreover, copepod symbiosis in a fry-containing pond increases oxygen consumption and reduces water quality. Therefore, before rotifers are added to the pond to feed fry, filtration and a water replacement process are typically executed. The filtration process enables the removal of large impurities and copepods, and the water replacement process entails the removal of low-quality water that contains a large quantity of ammonia toxins. These processes ensure that only rotifers and copepods with a size of 122 to 176 micrometers, which are suitable as a feed for a grouper fry, remain in the culture. All right. So, the system. Beto says, met Featherfin, the crypto dealer. That's what I'm saying, man. I don't know what's going on. I downloaded it, but my money hasn't hit yet. I emptied my bank account. What more do you want? This method is manual filtration. This method is used in commercial rotifer production and involves the use of two meshes with different densities. A 100 micrometer mesh on the first layer, which is used to block impurities, and a 1 micrometer mesh, which is used to harvest rotifers. This method is cost efficient and highly adaptable. Drum filtration. This method is the most commonly used mechanized filtration approach in high density aquaculture. In this method, a rolling bucket with filtration mesh is installed on a breeding tank to enable rotifer filtration and prevent rotifer adhesion on filtration mesh. A bucket type net system. In this system, a bucket type net is positioned at the center of the rotifer culture tank, and water is passed through the filtration equipment, including a sedimentation tank and protein separator. Subsequently, the water is transported back to the rotifer culture tank. This system reduces the quantity of wastewater and ensures high quality water for rotifer cultivation. That sounds like too much work. What the hell is going on here? For a cone type net system. This system comprises a cone type net that has a diameter and height of 60 centimeters and contains a 50 micrometer mesh. The mesh can hold 200 liters of rotifer. However, this system requires the use of handheld sprayer to wash off the rotifer that are attached to the mesh, a process that is labor intensive and cannot be automated. Trash. Dynasty America currently playing here in Seattle. Oh shit. I'm about to get off stream and watch that. That's my excuse. Come on. It's history. History in the making. Johnny Best said, my friend tries the same with me. If I send someone Bitcoin, they will triple X me back. Oh, hell no. Yeah. That's so crazy. Culture system with a membrane filtration unit, CMFU. In this system, a chlorinated polyethylene filter is installed in a rotifer tank. A peristaltic pump is used to pump seawater to replace the culture water, after which the installed filter is removed by performing a gas backwash. DP Exotic says, was Featherfin behind the Hawk Tua coin? Bruh. Alright. The source of food. The rotifers, this species they used in this study was Brachionis ibericus, cultivated at the National Shenkung University in Taiwan. The body size of the cultivated rotifers ranged from 105 to 250 micrometers. We used rotifer strain that was sourced from the Tungkong Biotechnology Research Center, Fisheries Research Institute, Agricultural Committee, and Executive Yuan of the Republic of China. This strain has been preserved in the Department of Biotechnology and Bioindustry Sciences since 2010. To stimulate a commercial breeding environment, natural seawater was used for rotifer breeding and was maintained at 25 degrees Celsius by using a heater. 25 grams of commercial algae powder, S-Sparkle from Culture Selco Technologies in Thailand, was added to each breeding tank 4 times daily after being homogenized with 500 milliliters of natural seawater. So they mixed it all up with it. The natural seawater used in the study was extracted from the sea off of Tainan, Taiwan, and salinity was measured daily by using a salinity meter and was determined to be 2.8%. Impurities and microorganisms were filtered using a 1 micrometer tube filter and stored in a 500-liter polyvinyl chloride PVC tank after ultraviolet sterilization. Oh my god, man, this is all for some damn rotifers. That's how you know it's gotta do something useful for the fish, because there's no way they're putting in this much work for something that has no benefit. You know what I mean? Like, this is like way too much work. It gotta do something. The rotifer concentration was measured using a dissecting microscope. The measurement process, they used a pipette, which the sample was collected and stirred, after which 1 milliliter of rotifer culture was drawn randomly from the sample and dropped onto a 24-well cell culture dish. Iodine solution was then added for staining and fixation, and then they put it under the microscope for manual visual inspection. Johnny Best said, Captain Critters, what's your website? I'll give you 2 guesses, Johnny. It's 2 words. One starts with a C, and the other one starts with a C. Oh, Captain put it in the chat already. It's captainscritters.com. Oh my gosh. Not a sponsor. Just a dedicated shill in the chat. Let's go. Therefore, the sample is... Sorry. The density of each sample was tested 10 times, and a standard error rate of 10% was achieved. All right. Let's look at the design. Enough of these words. So here how they designed this culture. GP said that's a crazy mystery. Yeah, what could it be? So this seems quite complicated. I'm not gonna lie. This seems quite complicated. So we have waste going out here. This going to the fish tank. We got a pump here. All right, filtered water coming up through here. Goes through filter A. The waste comes out here. This is waste 2. Then it goes through filter B. That goes out waste line 3. Then it goes down into the stay tank through another filter. A 50-liter tank. And we got breeding tank 1, 500 liters. Breeding tank 2, 500 liters. The design of the system is displayed in figure 1, as I just described. The system includes 8 500-liter breeding tanks used to culture the rotifers, and you use a 50-liter stay tank for holding the rotifers. So the breeding tanks are here, and we got them up in this stay tank as well. JPC, so that thing is a must-have to culture. If you got larval fish, super small stuff, I think a mono shrimp, raising a mono shrimp might be one of the uses most relevant to me personally. But beyond that, I don't have that many fish that are too small for like microworms. See, the thing is, I need to have something that needs a food smaller than microworms. Microworms get a lot done for me. I'm not gonna lie. I'm not gonna lie. They get a lot done for me. But I also have a lot of killifish and stuff that can eat brine shrimp as soon as they're hatched. So I don't deal with too many super small stuff. Probably the rainbow fish fry that I'm starting to get back into is the smallest thing. All right, what was I saying? Talking about the breeding tanks. The tank is equipped with 2 water level sensors, high, low, which transmit water level related information to a PLC implemented in the system. The number of tanks and size of tanks can be adjusted as necessary. Furthermore, the system has 2 filter housings, each of which contains 1 filter. The principle underlying the filtration process in the system is similar to the principle underpinning the manual filtration method. The filter in the first housing, filter A here. Captain Critter says, I'm biased. The info in my takes is going through Slasby believes rotifers are not worth it. I haven't convinced myself yet, I'll say that. But someone might have a use case for something this small, in which case it might be worth it. Maybe if you're doing any of those Amphidoris, mono shrimp, maybe Empire gudgeons, the fish that get the super, super small babies and have like 5 billion of them. GP says, Captain, do you think scuds would be a problem in a Daphnia culture? A Daphnia culture? I don't know. I'll have Captain answer that. JP says scuds will become a problem eventually in any situation. I got that. Mick said it's often easier to keep larger tanks or tubs when propagating live foods, easier to maintain proper water quality and accepting mistakes. That's true. Mick in the chat with his wisdom, even though he's tired. So, we have filter A, and in the second housing, so filter A is 100 micrometer mesh in this filter, and then filter B has the 1 micrometer mesh. Two manual valves positioned adjacent to the two filters enable the extraction of water from the first housing and second housing for sampling and analysis. For rotifer extraction, the electric valve at the bottom of one of the breeding tanks is allowed to open to the rotifers to flow into the central pipeline. Subsequently, pumping is executed, pushing the rotifers upward. Numerous valves control the direction of rotifer flow. After the activation of pumping process controls the flow of rotifers into filter systems. TP Cell, hope you back. I'll hop off because the names that want to know about a broken AC in my work area. Oh, man. Lost to Finn America said, I keep scuds and Daphne together, but I don't keep scuds with my cherry shrimp and isopods. That I would agree with that decision. So here we have a picture of the actual system, not just a diagram. So here we have the pump. We got the sample 1, sample 2. They're going up here through filter 3, filter B, filter A. They got another filter on the end of that into the stay tank. And here's one of the breeding tanks with the high water level sensor and the low water level sensor. So honestly, it's not that like looking at this. Now that I've finally, put my water through a sediment filter and a carbon block, which was stupidly easy, and I love these. This is the best thing ever. Just a little, what is it, 1/4 the RO tubing. I mean, it's not that hard to push some water through these kind of filters. So honestly, it wouldn't be that difficult to set up something on a smaller scale like that. But obviously, this is massive. I don't think this has none of the things we've gone over are really practical methods for culturing at home. I mean, it'll probably just look like you getting a tank, putting rotifers, or a container, putting rotifers in it and feeding them phytoplankton or yeast or whatever. Mixed, I failed to keep ammonia so many times when recommended to keep them in jar-like conditions. I know. What keeps happening is I have them and they're going great, and then I have to like leave, and I haven't hit that like perfect point in a while where they stay around forever. I keep fumbling in between like doing stuff. I need to get a tub outside for them, although it is really hot. But that's why I've been thinking about getting a tub outside and doing Daphne in it. But I'm just worried about contaminants, and then I have to manage the thing outside and the heat and not making mosquitoes. And I just don't feel like dealing with that. I really don't. Okay, continuous filtration. Got it. Man, we got rinsing times. Yeah, we got to let me get to something more practical again. This is cool, though. The whole point of this is we're trying to learn what rotifers are and their use cases and decide if we should actually use them. Now, let's go to this next paper. This is another aquaculture journal. I'll put the link to this in the chat. Captain Critter said, Aquarimax Pets ran experiments. Scud helped Daphnia cultures by ripping up and keeping them suspended while having snails lowered yields, but overall Daphnia by themselves did the best. Okay. Makes sense. This is the suitability of different live feed for first feeding of freshwater fish larvae. And this is where we get into some of the comparisons, or just a quick overview of them. Species involved in the study. Okay, we got some different stuff. They did paramecium, microalgae, paramecium, and rotifer. Starter cultures of P. bursaria, P. pulchatilis, and B. calliiciflorus were obtained from commercial suppliers. They fed P. bursaria with bacteria. Bacterial cultures produced using rice/alfalfa medium, and P. bursaria and chlora sp were added to the culture substrate. They had Artemia cysts up in here, and they did some larva first feeding experiments So let's see what happened. We got Artemia, Paramecium, rotifers, microalgae. This is the most you could ask for, really. All the different foods. Previous studies demonstrated that S. Lucioperica and L. Lata larva. Let's see what this is. A pike perch. Okay. Sander Lucioperica and L. Lata fish. Okay, Lata, the burbot. All right. There's so many fish out there in the world that people be eating and culturing that I'm not even familiar with. It's kind of crazy. I feel like the fish offered to me are so limited. I need to be trying more fish. I mean, I had monkfish at least, but it's like what? Salmon, tilapia, catfish. I need to expand my horizons of fish that I've been eating. Diana said, my Daphnia and Moina are struggling. A video said to put snails in. I'll take them back out. All right, I'll let Captain respond to that. Expert on that topic. So they found that at the larva stage of fish first feeding, they're sensitive to manipulations necessary in fish culture. Lowering and increasing the tank water is required for cleaning and stuff can disrupt buoyancy or injure the larva due to turbulences and mechanical forces. Therefore, in their first feeding experiments conducted, they were in rectangular tanks with volumes of 200 liters under static conditions without aeration and water renewal. Aeration in tanks stocked with L. Lata and S. Lucioperica were started when oxygen saturation dropped to 75%. C. aternensis larvae were kept in similarly sized tanks. And that is another big fish. Hey, Wikipedia don't even got a picture of them. What's going on here? Alright. Big shiny fish. Fish were kept on live feed for 20 days from the first day of feeding, as these time periods were considered suitable to detect differences. Let's see. Fish suffering from malnutrition show either heightened mortality or significantly decreased growth rates at this time. Loss of fish due to cannibalism might falsify feed-related survival rates, but during this period, no cannibalism was observed. All right. Fish investigations. For determination of survival rate, all individuals were connected with a tank with a fine meshed hand net and counted. The survival rate was calculated as the number of larvae surviving at the end of the experiment in relation to the number of larvae stocked at the onset of experimentation. They also looked at body length, body depth, and body mass. God damn it. They always catch me out surprised, man. They kill the fish at the boat. I didn't expect this to be the part where they talk about killing the fish. Like, you tell me if you saw this coming. What's up, Parna? Parna said, how easy / plentiful are backworm cultures? I've been hearing a lot of people say it's easy. I would say people seem to do it. I haven't personally done a bunch of it. What's up, Mexicali in the chat? How's it going? Swim Deanna Jones. Long time no see. And Captain is linking the Discord if you have any questions about Daphnia and things like that. Captain is in the Discord. Captain Critter is like one of our only YouTube mods and one of our main mods in the Discord. Although SVC, shout out SVC, when Ben's account got hacked and he was posting some crazy stuff in there. I think SVC was on top of it. Shout out SVC. All right. We have the best mods. That's why I only need like 3. So, tell me if you saw them killing the fish coming in the sentence. For the determination of survival rate, the survival rate was calculated as number of larvae surviving at the end of experiment in relation to the number of larvae stocked at the onset of experiment. For determination of body length, body depth, and body mass, larvae were killed by prolonged exposure to 0.2% of MS222. So this is the same thing they killed the fish in with the other paper. 30 larvae in total were randomly sampled at the onset of the experiment, i.e. day 1, and 30 larvae per tank, 90 in total, at the end of the experiment. They were photographed in a stereo microscope, Visioscope SZT360 coupled with a video camera, VisiCam 3, or with a cell phone camera, Galaxy X Cover 5, depending on their size. Partner said, I mean, they need to kill them to sample them, right? Yes, apparently, yes. But for some reason, no matter how many of these we read, it always catches me off guard. I'm just reading, and it's like, the fish will then humanely demolished. All right. Where was I? There we go. All right. Larva body mass was determined using an analytical balance after the water had been removed with tissue paper at a precision of 0.5 milligrams. At day 1 in El Lada, body mass could not be determined as larva fragility due to the small size allowed no accurate analysis. Skeletal malformations were determined at the end of the experiment on the 90 sampled larvae. Larvae were fixed in 4% buffered formalin and stained with Alcyon blue and alizarin red for cartilage and bone. Beautiful. Beautiful. Larvae were photographed with a cell phone camera, and pictures were analyzed on the following malformations potentially occurring in the fish larva. All right. So let's look at the results of this. Remember, we're looking at the results of the feeding trials. We got Artemia, Paramecium, rotifers, all the good stuff. The fish were humanely put in a sock and thrown against the table. No, they used MS222. Come on, man. That's the most humane thing on the commercial market. So here we have some pretty cool photos. This is figure 1. Figure 1A shows L Lada magnification x25. So that's the fry right here. B is detail of its head. And C, we have the other fish, S. lucioperca at x15, and then we have the detail of its head. So they're looking for any malformations. So they're taking a picture of that. And you can see some malformations in figure I right here versus the normal vertebrae below. So you can see the picture below in J. You can see that the spine is like more evenly sized, straighter, and then in I you can see that it's kind of weak in the middle here. Then in G we see a normal jaw, and then H we see a larva with a prolonged lower jaw. Captain's Critter says MS222, also known as tricane methane sulfonate, is a chemical agent commonly used in veterinary medicine and research for the anesthesia and euthanasia of cold-blooded animals such as fish, amphibians, and reptiles. It functions as a sodium channel blocker which disrupts nerve conductivity and leads to loss of consciousness and physiological activity. Wow, loss of consciousness and physiological activity. I'm gonna have to use that one. Okay. Live feed. Morphometric characterization of tested live feed. The fraction of sieve netted zooplankton used in this experiment was 100 and 200 micrometers in size. This contained 51% copepodites in the CI stage, 56 napuli, and 3% freshwater rotifers. All right. Feeding trials. Here we go. This is what I was looking for. Feeding trials. Survival, total length, and body width of S. lucioperca larva was similar when fed with the feeding regime A or C, A, B, or C. Let me go back to the feeding regimes because I think I missed feeding regime. Section 3.3. Feeding regime A. Okay, the feeding regime used Reading is so hard for me right now. Not gonna lie. USA, JP said. Yo, how's the game going? So this is the feeding machine. There we go. B. calixiflorus. So this is the rotifer. Alright. B. plictillus. That's another rotifer. And P. brusaria, which is a species of ciliate, famous for its mutualistic endosymbiosis with Zoochilura green algae. Alright. So they found those three were suitable for the first feeding of S. luciaperca larvae, as survival rates were high, 80%, and significant growth, increases in total length of 50% and in body length width of 20% was obtained. The nutritional content of rotifer species has been discussed in several papers. The concentration of highly unsaturated fatty acids, their respective ratios, and the vitamin levels are limiting parameters of nutritional value, but they can be modified and improved by different enrichment procedures. Although no nutritional information is available for P. brusaria, it's considered a valuable light feed as one P. brusaria cell contains up to 700 symbiotic chlorella sp cells. These symbiotic cells supply the host with photosynthetic products, namely maltose, while the host provides the algae with nitrogenous compounds and carbon dioxide. Paramecium caudatum is a food organism that is less elaborate to culture, as only bacteria are necessary for its culture. It was successfully used for first feeding of the larvae of P. fluviatilis and of the S. luciaperca hybrids. However, the culture water of P. caudatum contains low oxygen and high ammonium concentrations and is inadequate for fish larvae. Under practical aspects, the utilization of freshwater live feed has clear advantages for freshwater fish larvae. The organisms survive in the fish tank for unlimited time periods, and no salts are introduced into the tank, resulting in stable tank conditions and water quality. Artemia napoli and copepod napoli/copepodites were not suitable for first feeding of pike perch larvae, as survival rates were low and growth was heterogeneous. Morphometric measurements showed that the Artemia napoli were too large for pike perch larvae, while they used lake zooplankton was smaller and might be ingested more easier. However, the locomotor activity of the prey also plays a role for the capture and ingestion success. Copepod napoli and copepodites have a specific motion consisting of sequences of sinking, swimming, and jumping events, and they show escape reactions in response to predators. This might reduce the capture success of larvae in the first feeding stage. In nature, ciliates can contribute to 40 to 60% of the total consumed carbon biomass of S. luciaperca larvae, and this is similar for other Percidae larvae. In turbot, Picetta maxima, capture success of copepod napoli was greater than 50% during the flexion stage and increased to 73% in the post-flexion stage. In contrast, Artemia have no effective escape responses. Beside capture success, the biochemical composition of the prey may play an important role for the performance of fish larvae, as it differs significantly between ciliates, rotifers, and copepods. Moreover, copepods contain a chitinous exoskeleton, which may be difficult to digest by larvae. The present study demonstrated that the aforementioned feed items used after the initial 10 days had a significant influence on larval survival rate and growth parameters of S. luciaperca. Artemia feeding resulted in significantly lower survival rates and in more heterogeneous growth than copepod feeding. Low survival rates and heterogeneous growth are disadvantages in larviculture, and they reduce the number of produced fish and favor the development of cannibalism, and require sorting/manipulation procedures of larvae/juveniles in early life stages still sensitive to manipulations. Low performance of S. luciaperca larvae fed with Artemia napoli may be due to its larger size, as discussed. Additionally, the nutritional profile of Artemia may be suboptimal. Performance of S. luciaperca larvae can be improved when Artemia is enriched with highly unsaturated fatty acids, etc., etc., etc. From this data, it becomes obvious the development of freshwater copepod culture systems is an important goal for the improvement of freshwater fish larva culture. Independent from the feeding regime, a percentage of 10 to 20% of pike larvae revealed malformations in the present study, predominantly lower jaw elongation. This could be related to the pike perch strain or to rearing conditions, and further investigation are necessary on this topic. A switch from initial feeding with the freshwater live feed of the rotifers to saltwater on the Artemia resulted in decreased survival rates and is therefore not recommended for hatchery practice. The exact reasons are unclear and might possibly depend on taste changes. Results of comparative feeding experiments on L. lauda larvae were mainly similar to S. lucioperca. Feeding regimes of the rotifers were preferable. Rotifers and copepods were preferable to the feeding regimes consisting of B. picotilis and Artemia napoli, and of B. calyiflorus and Artemia natuli. Okay. Whew. Reading, man. Reading. Human workload and potential cost for production are relatively similar for B. calyiflorus and B. picotilis and P. busaria. Microalgae must be cultured as food organisms for rotifers and as symbionts for P. busaria. Further, it is necessary to maintain stable water quality in the rotifer and protozoan populations to avoid their collapse. Artemia production is less elaborate, as it requires no food organisms. Culture techniques have been described in detail. Collection of copepods from wild zooplankton populations is a special technique with several requirements. The fish farm has to be located near a zooplankton source to avoid long transport of food organisms, and adequate equipment and permits are necessary as well. In the required densities, copepods can only be stored for limited time periods of approximately 24 hours, therefore regular collection is necessary. Alright. Another important aspect that needs to be considered with live feed is their role as potential carriers for pathogens. Both cultured rotifers and Artemia have been shown to be potential vectors, and nevertheless, as these organisms are grown under controlled conditions, adequate hygiene concepts can minimize the risk of pathogen transfer to fish larvae. In addition, copepods collected from wild populations may be source of pathogens themselves, and it's well known that distinct copepod species are fish parasites or serve as intermediate hosts for parasites. On a practical scale, copepods collected from a lake population have been used at the study of the fish farm for intensive culture of several million larvae of different freshwater fish species for decades. According to unpublished hatchery protocols, copepod feeding was never associated with the outbreak of massive fish disease. However, there's a risk that distinct percentages of fish becomes infested with specific parasites during particular times of the year. That's one thing that's interesting. Some stuff is kind of seasonal. Like, I know there's some places where like you don't want to order, or it's not even legal to order fish from that time of the year, but even if you could, you wouldn't want to, because during that season there's a lot more parasites and stuff afoot. So in conclusion, they concluded, the rotifers were suitable for the first 10 days for this S. lucioperca, and that the same 3 types of live feed as well as lake copepods were applicable for the first feeding of L. lauda, and that they were seemed to be superior to Artemia. Whew. There we go. I'm getting a clearer picture here. It may be if you're doing something in mass on, you got, if you're J.P. Jeffries and you got 500 rainbow fish fry, maybe you'd benefit from a rotifer, culture. JP is now our stand-in for big aquaculture. All right, how we doing? Everyone's still alive? Holy. Let me take a quick pause here real quick from the reading. Let me take a quick, but I'm just checking in to see if you're all right. Captain said, still alive, still unconvinced, still biased. Well, I mean, you don't really have any use for rotifers. I feel like you've got all the different foods that you need already, most likely. So I understand that, but you never know. Someone out there might hear this and they might be like, okay, this could be good. I got some green water. Let me get some rotifers. Let me feed my rainbow fish fry or whatever super small thing I got. What's the problem? Captain says, but I've tried to keep them 4 times without success. Oh my gosh. Oh my gosh. We might have to do a part 2 of this because the fatigue is hitting me like a train right now. But I still got so many more. I got so many more papers loaded up. Ugh. Oh my gosh. I'm just too tired. I'm just too tired. Well, tomorrow I don't have to wake up super early, well, as early as today. That was the thing. Waking up today and trying to get all my packages out. That's what killed me today. With the energy levels. Smartphone said, alive, just listening while I sand this board, hopefully staying in seal tomorrow. Okay, I'm hoping I can stream tomorrow too. By the way, I got some good feedback on the last stream that we had. I know it seems so long ago. But the last stream we had, it kind of came about randomly. And I appreciate the people that like messaged me, and then also some people left comments. And that was the video where I went over how the Aquarium Store Depot channel seemingly stole the Koi Pit's entire video. They just completely copied the script. And shout out to Fathom for sending the video to me and being like, yo, did they just copy this? And I went and did the transcript analysis or whatever. But I had a couple people message me, a couple of comments, people saying they unsubscribed from him. Just, it was a cool thing to catch, because I think it's important to call this kind of stuff out. But also it was just good to see that people appreciated it. Captain said bonkers, GG, the Koi Pit, and Fathom. Yeah, that was pretty crazy. I saw, we can fangirl. We got freshwater ichthyology. She commented, she said, Aquarium Store Depot has some really odd videos. I watched a few and not surprised there's stolen content. Pond Life Aquatics is one to check out, as their nitrifying bacteria one is very similar to Alex's similar video, but also seems to have heavy use of AI. Definitely for the thumbnail citation references, but likely the script is AI. Then shout out to my lurkers. I need to respond to this comment. Laura Victoria Tanks, she commented, said, hi, hey, I'm mostly a lurker, lol. Just want to say thanks for sharing. I'll be avoiding Aquarium Store Depot's videos from now on, because that's no okay. If you're a lurker, you just watch the replays, shout out to you. No pressure to ever comment or anything, but I do really appreciate a lurker popping out and being like, oh shit, that's someone I've never seen their name in the chat, never in the comment section, but they're out there. I'm just gonna have to have faith you guys exist. I see, there's a good population of people that they don't catch the live streams, but they do watch the replays. So I always keep that in mind. That's why I always try and hide the amount of people watching me at once, because I'm like, look, if they're not here now, they'll see it later. It's kind of my goal. To Platypus said, L AI Depot, L Prestige Aquatics, W Big Steve, W Stream, and Aquarium Domain said, great stream. Shout out to Aquarium Domain. Yeah, I remember Aquarium Domain. This is from our perspective, a pretty big channel. I remember once he gave it the live stream, and it was when we were talking about the list of bald YouTubers with beards. I remember he came in a live stream chat. He was like, "What the hell is that you talking about?" Yo, you never know. You never know what we get up to over here. You just never know. So, anyways, that was a fun stream. That was a good time. I feel like we've been streaming consistently, but the it needs to increase. It does need to increase how often I'm streaming. It's just become more challenging. But damn, we have streamed a lot in general the last whatever months. Hours and hours and hours of live streams. But I still think there's more work to do, for sure. Before I get off, anything you guys want me to take a look at? Anything? Any cool videos? Any news? You want to instigate anything? Come on, now's the time. Put it in the chat. Whatever. You have my attention. Because if not, I'm out of here. Let's see. I'm gonna search aquarium in YouTube and set the filter for this week. What came up? Oh God! Look at this, 200,000 views in one day. Buying monster fish for 2,000-gallon aquarium. Yeah, this looks like some slop for sure. I built a natural ecosystem in my aquarium. No more water changes, no more algae, self-sufficient. Oh, is this that other AI channel that's been popping up recently? It's like Skeptical 2.0. Captain Critter said for the memes, baths versus showers. Like, you're asking me to give my opinion, or is there a video?
Sounds impossible. I have not done a full water change on this tank in over.
Yeah, the Aqua Tank Tony. This is AI. Oh wait, it has an AI thing on it.
I want to tell you something that's.
Look, it's self-described as AI. That's nasty work, bro. At least they tell us. At least they tell us. The best way to mount your aquarium heater. Pause. Which aquarium substrate is actually better? Oh my God, man. Decisions. 8 months with my Indie Aquatics aquarium. My honest review.
Had my 5x2x2 Indie Aquatics tank for around 8 months now, and in this video I wanted to give you my proper honest review. Now, just to be clear guys, this video is not sponsored. These are my contrast with the woods and the plants.
Oh my God, that fish looks massive.
And I also have a hood, and the cabinet has holes drilled in the back for the filter tubes. Now, the ordering process and communication is amazing. The team is so helpful and they're very knowledgeable. Like, one of the main things I was worried about was the delivery was about 10-20 minutes max. Like, it was so quick. Now, let's talk about my first impressions. And the first thing that went through my head was just,
guys, rate this scape. Let's go out of 42. Rate this scape here. By the way, shout out Scoobs and Pets. I think we watched his videos on here before. He seems like a cool dude. Let me just get that out the way. What the hell's going on with this tank, man? Captain Critter said, ooh, Thumbnail Wars episode 2. Yes, I do want to do a thumbnail. I was actually thinking about that the other day. Big Steve says, speaking of nasty work, where's the degenerate Bomba been? That's a good question. Bomba been MIA, man. I haven't really seen him. I think he's been busy. He's a working aquascaper. He's still aquascaping though. He was just posting something he was working on.
Wow, this tank is beautiful. It was.
I don't know, man. I'm gonna keep it light, but this tank, you could use Bomba's help. It's a little uninspired. I don't know if this little plants in the back just aren't, tying it together for me. Something's just not connecting. I like the wood. This wood piece, we're under a good start here. I mean, this is probably right after he set it up, so it's a new tank. Needs more plant. You rated 20 out of 42. Needs more plants. Yeah, I think any tank that's super empty and that has this massive fish. Now let's see. There's gonna be more plants. It's a 90-day thing, so let's see how it progresses.
Was just wow. I mean, I've got my Fluval Roma 240 in the living room as well.
Now this is a different tank, but this looks nice. I like this. Got a little. Okay, we got some plants. We got wave maker.
And it was directly opposite the ND Aquatics tank, and the
Was that snakehead?
difference in the way they looked was
Is that?
just
Is that some kind of gudgeon? I can't tell.
The ND Aquatics tank.
A couple fish in every tank needs more plants. What catfish is it? It looks massive. It looks like it's gonna outgrow this tank in like
Looked
a month.
so much nicer. Like the tank looked premium compared to my Fluval Roma 240 or any of my other tanks. I mean, even here, the Interpet here, there's a plastic top. My Jewel 110, which is out of shop, but you can hear it. It's a plastic top as well. And yeah, like the first thing that stood out to me was just that premium look. The ND here, we have the woods and the plants that allows the smaller fish to hide.
Okay. We're getting some more plants in here. This is looking better.
A little bit. But anyway, that's the fish in that. We're actually talking about the
Diane said 35. I think that was for the last one. Captain said look premium, water hella cloudy. I mean, you just set it up.
So the magnet, this is the Hygar magnet. The good thing about this magnet is if you accidentally lose it, it actually floats at the top, and then you can just get it back here like that. And yeah, that is the best way to kind of clean the glass. It works fine. Now I should mention that the Hygar magnets, they come with blades. And then we've got the heater, which is reaches all the way to the other side.
I just want to see.
Anyway, that was a closer look of the tank with the way that.
Okay, this is looking a little bit better. We got more plants. Okay, look in that back corner. That's what I'm talking about. This is we got some roots exposed. I guess this catfish is probably digging up some plants or something.
Hardscape came out. One thing I should mention though, definitely pour all of your hardscape and stuff in before you add in the hood, because once you add in the hood and the glass panels, the openings for getting stuff into the tanks are of course much smaller. But yeah, in overall, I'm gonna be honest, there are definitely
it's a very nice.
I spoke to a few. It's all high quality
tank.
Of place.
Does look very fancy. I think I would have done something different personally, but you know, it looks cool. It looks nice. Big fish. Baths versus showers, though, you gotta have both. You should take a shower, then take a bath, then take another shower to rinse yourself off after the bath. You gotta have both. It's not a either or. It really isn't. I'm sorry. This is not possible. If I could only pick one, it would have to be showers, because baths is a luxury. I don't have time to take a bath every time. You know what I mean? So I would have to choose showers, just like you can't just take a bath casually before you're getting ready to go in the morning. That would just add too much time. If you're a nighttime shower, before you go to bed, you might not necessarily want to do that. How many people are in nighttime showers versus morning showers? That's a good question. It really depends on what's going on for me. We do have to do another thumbnail war.
Before aquaculture,
the problem is that the AI is really making the thumbnail rating difficult. Because I don't want to rate a bunch of AI thumbnails. You know what I mean? God, look at this. This guy looked like he about to get you killed on the job site. No cap. Our most dangerous installs, massive aquarium. This guy looked like he definitely gonna get you slaughtered on the job site. Day 1 to 45, no filter aquarium transformation, step by step. Sparkling gourami tank. I'll be honest, guys. Damn, we got the Windex glass cleaner. Wait, why are we doing the Windex like this? Oh, for the black film. Okay, I'm like, what the hell's going on? All right. Okay. We got the black background. We got a yoga mat. Man, I can't watch videos like this. I'm not gonna lie. Well, look, I don't need to see him fondle the stick for this long. Look, what? This is some. What is it? This is some indie fish tank shit right now. Man, we got a cup to pour the water into. We babying the substrate. Nah, just dump the whole bucket in. Oh my god. All right. Smartphone said these are like cooking videos, not about how you cook, just to watch someone else. Yeah, I can't do it. I really can't. Thailand's biggest aquarium fish show. Oh my gosh. Look at that. It's gonna be a bunch of big ass fish. Ooh, we got aquascapes. Now this is what I'm talking about. Oh, we got a little figurine down here, a little cat action. Okay. Yeah, I gotta get out to Thailand, man. They're doing it right. Look at this. This is peak right here. Bomba, get on it. Oh, now yes, more neon tetras. Give me more neon tetras. Let's go. Are those threadfin rainbows? What funky ass combination is this? All right, hold up. You may have lost me. What's going on here? Neon tetras and threadfin rainbows. I don't know how I feel about that. Okay. Guess what fish are in this tank? You got it. Neon tetras. A green Kubatoy reservoirs. Damn, look at the plants though. Oh man. I like them dripping downwards. Now this I can look at and appreciate. I like how these are using like, you know, sculptures and stuff in them. It's not like, and there's a lot of shrimp. There's a lot of red shrimp in these. Got cardinal tetras. It's like they only use 2 fish, man. What's going on? Partner says, what's the probability of my parados eating Histatus eggs if they spawn? I feel like you know better than me. I haven't done that combination yet. I don't know if it's terribly high, like, that they get all of them. Oh, look at this one. Look. We need more aquascaping competitions like this, where it's not like some ultra Apple Store aesthetic. You can also just use props and things. I like this. Like we still got the glass pipes and stuff. But look, we got a little mannequin here. Got a fence path. Like this is good. This is good. Yo, shout out to Fathom, W Fathom in the chat, man. The FBI fish man says a member for 2 months said need more Bashirs in aquascaping. Thank you, Fathom. Thank you. Partner said there's an aquascaping comp here in Brazil who gave 4th place to an African cichlid tank that had nothing in it but sand. You hate to see it. W Fathom in the chat. All right, what do we got in this tank? We got some super colored down chili rasbora type thing, some orange shrimp. The stocking is always the worst. These fish look like they're gonna die in a second. Well, just they don't look very happy. Is what I'm saying. They look like they're getting gassed to hell. Look, I can see the bubbles everywhere. Yo, is that the CO2? Yo, what? They're putting it through a CO2 turbine. Oh my god. Yo, get the fish some air. Oh my god. The fish aren't supposed to be pearly. Look at this diffuser in the corner, man. Sometimes they pour soda water into tanks to act like it's purling. Yeah, I mean, I can see it coming out the CO2 diffuser, so I know that's where it's coming from. Okay, we got some more. Man, these fish are in awful shape. Look at this, man. At least one of those reservoirs. Yeah, this one up here. That one doesn't look too good. Oh, that one looks kind of funky. The wrong kind of pearl, Daniel. Celestial says they're going to be sent to heaven. Yeah, what kind of... But these Harlequin rasboras? The color in this one just looks so odd. I don't know if that's just because it's like about to cease its physiological function. However they said it earlier. Got a little elephant here. Ooh, okay. We got a more center focused. Couple fish said big bubbles are ineffective at gas transfer compared to small bubbles. So what does that mean in terms of the fish's survival chances? Honestly, these shrimp kind of look too big for this scale a little bit. I don't know. Got some more cardinal tetras. I like this, but the branches are kind of too thick for me. I don't know. It's cool. The scale is just a little off in a way I can't describe. Smart person says CO2 doesn't directly affect the fish like that, right? It's more about they don't have O2. Well, I believe the CO2 makes it harder for them to absorb the oxygen. So I think that's where it comes in. Nick, we need a stream on fish respiration and gas exchange. Oh boy. You guys know I'm a terrible educator, right? You've seen my reading skills. Are you sure you want more misinformation? Ooh, look at this one. Oh, we got a little entrance gate at the front. Oh, this is a cool... See, this is why I like the props. Allowing props in these, because we got some big-ass boosts over here. We kind of got the valley effect. Dark in the middle, bright in the front, bright in the back. And then I like the perspective play here. We have the gates right in the front of the tank. So it's like you're looking into the distance at this house back here in this little... A couple fish said now without it replacing O2 at high levels, fish don't know the difference. All right, I'm gonna look at some of these. What's it called? The Dutch style takes. You just see the fish dying before your eyes. The shrimp look great. A couple fish said if you have good O2 and the right fish, you can dose up to 600 parts per million of CO2, which is okay. But that's if you have good O2. This is my favorite one so far. I think this is a really cool scene. The shrimp are just kind of breaking the perspective a little bit. But besides that, because they really do a good job of making this house look kind of far away. And then you see the shrimp next to it, and it looks like massive still, you know. Yeah, that's a cool one. And you know another thing that's interesting about this is so much moss involved. This is all looks like all moss over here. Or is that like a pearlweed in this a little bit? Yeah, I like this. This is just infested with great looking shrimp. That's all I can say about this one. The escape is kind of a mess. I'm not really sure what I'm supposed to see here, but the shrimp are everywhere. We got some arowana, okay. Some red arowana. Big fish. Flower horns. Man, look at this monstrosity. This is such a weird... I do get flower horns because they just look so unique, but something about them just disturbs me. I don't like the brain head thing. It just looks... Oh, not the goldfish. I can't do this. Oh my gosh. It looks so gross to me. Look at this thing. I can't even see its eyes. Oh my gosh. Does it actually just not have eyes visible? Oh my god. What? Goldfish people are sick, man. What is up with this? It's just a floating ball of flesh. Can't see anything barely. Jesus. I don't get it, man. I don't get it. Steve said that's an abomination. Yeah, I don't know what the hell that was. These fish look jacked up. That's all I know. Oh, we got some discus. Okay. Oh, here come the bettas. Let's look at the bettas. Mini Scenes GB. So good man, just found you, and what I've seen so far, poor fish. I agree. I'm off to bed now, but I've subbed and will watch properly tomorrow. Appreciate you. Have a good rest. Thanks for stopping by. Always love it when people just, tap in the chat, even if it was for a second. I do like the white bettas. They are very appealing to me. Okay, we got a nice yellow betta. This is the level of my analysis here. The betta is yellow. It's not really sparking anything in me. Got a red betta. That's cool, I guess. Now, would you call either of these a koi? I don't know. It's got a kind of crazy combination of colors, though. Looks like a damn starburst on the right. Sometimes that fish on steroids. Ooh, now this is nice. We got a white body with red fins. I like that. Probably trimmed them to be perfect or something. Jose said, hello, Nick. Everyone, tank maintenance lurking. What's up, Jose? Yeah, we got some bettas. Man, oh man. Jose said those bettas are amazing. They look pretty good. They had some discus here too, Jose, but I don't know anything about... You tell me how these discus look. I don't know anything about discus. Got a blue guy. Yo, why he got a butt chin kind of. Got one of the red ones. I don't really like these solid color red ones as much as when they got like the patterns and stuff. Oh, this one won an award. Oh, that's really red though. His body shape's kind of crazy looking too. Interesting. Oh, we got this one that's all... Ooh, this crazy pattern on this discus. Look at that. See, I can appreciate that. I can appreciate that. Damn, there's some big fins on this betta. Look how much fin this has. Oh my gosh, it really got the wedding dress effect. Jose said, I saw the discus was just looking all amazing. Yeah, this is some crazy looking betta fish. This is with some damn crocodiles. What in the world? We barefoot with the crocodiles now? Oh, hell no. Oh, hell no. Captain said both beautiful and sad, JP says, I have 275 XP. Can I turn it in for the RC truck? Yeah. Just deposit some crypto into my wallet. I'll give it right back. Okay. I don't know what's going on there. What in the tornado of bubbles? Wait, did anyone see that? What are they doing to the fish? You can't even see the fish because of the bubbles. Is that in the same, actually the same area as them? Captain said, I had to dip and get dinner started, but it was fun hanging out. Yeah, it's nice having you around again, Captain. Thanks for stopping by. This is a pretty cool aquarium show, though. Not gonna, there's a lot of diversity and things to see. We got reptile stuff, too. More goldfish. Oh, here's the stuff for sale. Ugh. Mollies. Just a bunch of live bears. Oh my gosh. Oh, they're just scooping out the goldfish. Oh my goodness. Yo, this chick is pushing her luck here. Oh, the net broke. Let me zoom in on the net. Let's see. That's crazy. JP said, I think that's a food market. I don't think they're eating the goldfish, bro. No, I don't think so. They're just, look, what is going on? Maybe it is. They're putting them in a pot. What the hell is going on here? Bro, the net broke again because the goldfish are so damn fat. This is crazy. What in the world? Jose said, stop staring, Nick. Yo, don't project your dirty old man tendencies onto me, Jose. I'm looking at these two beautiful goldfish here. Why did you give her another net? You're just gonna break this one again too. Oh, this is disgusting though. Look at there's something about them all being crowded in this pot, just like flipping around. Like, this is terrible. Wait. I think that might be the point of this, to see how many you can scoop out before the net breaks. That might be the point of this, because I feel like they're counting how many fish. Yeah, I think that might be the point. Ah, okay. So you're supposed to see how many you can get before it breaks. I think is the point. That makes more sense. Fish and Jay said late to this one. We're wrapping up, but you know, always in the stream by looking at some horror shows these days. Ew, god damn. These goldfish are so disgusting. Oh my gosh. But even they don't deserve this treatment. Like, what's going It's just like a game where you try and see how many you can scoop without the net breaking. How many times you gonna try this? Okay, one. Christian J said they look like high quality ranchu. Just scooping them in the pot with barely any water. High quality ranch. Ew. Yeah, I can't do this. Fish said at Luke's Goldies. Lostafin said going to take a shower because they're better than butt water baths. Night guys, yo, what? How dirty are you guys? Everyone keeps saying that the baths are like dirty water. You're supposed to wash yourself before you take a bath. I don't understand why this is so complicated. Also, if you're worried about butt water, your butt is on you 24/7. So, you know, you're probably getting exposed to it more at that swamp ass than by taking a bath. Inkji said bedding. All I know is this is fish abuse. That's all I know, but I don't think that they care. And honestly, I got bigger fish to fry, no pun intended. Alright. Well, with that being said, let me make sure I cleaned up all that water that spilled earlier. Let me get to going, get to feeding, get to doing all that stuff. And yeah, hopefully I can be back ASAP. But it was nice stepping in. We got a little bit further on understanding rotifers and different use cases for them. I think there's probably still a little bit to do there. But one of these days I'm gonna do like a catch-all stream where we just kind of go back to any topic and read a bit more on it. I never feel like I get far enough in these. But hopefully you guys found this interesting, even if it's not useful. Or you did find it useful and interesting, whichever one. And I appreciate people holding it down in the chat heavy tonight. Jose, Diana, Inked, Lost a Fin. I think I saw in the flesh in here. Big Steve, a couple fish, of course. Parna was up in here. Parna up in here, smart person. Good. Hope the construction's going well. Captain's Critters, always nice to have Captain in the chat. DP Exotics holding it down. Featherfin trying to hack me. Not through the YouTube chat, buddy. I'm safe. Beto, Mickim, of course. Johnny Best, thank you for the membership. Fathom, thank you for the membership as well. Appreciate you. We'll look about the Bashirs and the thing. The Koi Pit stopping by. Appreciate you. Ben, come on, man. What's going on? Where you been? No pun intended. Jeffro, appreciate you stopping by. Holding it down the chat. Savage Cicklage. Jonathan, I know you're up in here. Yeah, there's In the Flesh. In the Flesh was up in here, too. Ross had been lurking. Appreciate you, Ross. Appreciate all my lurkers, as always. Even if you're just a lurker, you know, you don't have to leave a chat. I know you're there. You know you're there. That's good enough for me. Andy Carson stopping at the end said, thanks. Appreciate you. I do like, you don't have to say anything, but every now and then, some of the lurkers I've seen they'll say something like Andy, they'll say at the end, just thanks. And I know they've been there. So, oh, okay, that's cool. Or some of them will leave a comment, but it'll just be like a thumbs up. You know what I mean? Like, hey, I was here. So I appreciate all that. It lets me know that people are enjoying and I should keep streaming. So it does encourage. JP says collecting from Poly Market. Yeah. So I appreciate you all. Leave a comment if you have something to comment on. You have an opinion on something we went over. What do you think? Rotifers, is there some potential there? We probably need to look at some smaller scale operation examples, but let me know. This has been In My Tanks. I'm going to go now. And I hope to see you again soon. And yeah, like I said, this is In My Tanks, and I'm out. What do you want me to say? All right, I'm leaving. Is that fine? Can I leave? Am I being held against my will? Is this a kidnapping case? Because I got it all on camera. This is all live stream on some poo shiesty shit. Like, you're not gonna win. You're not gonna beat the case. All right, I'm sending you straight to jail. So this is In My Tanks. Don't try and kidnap me. Don't try and hold me against my will. I'm out.